What are breakthroughs in lymphoma genetic profiling? Imagine a world where doctors can understand lymphoma at a level so detailed that treatments become more precise and personalized. Recent advances in genetic profiling are making this a reality, especially for non-Hodgkin lymphoma, which is a diverse group of blood cancers affecting lymphocytes. These breakthroughs are helping scientists and clinicians identify specific genetic and molecular features of different lymphoma types, leading to better diagnosis, prognosis, and targeted therapies. One major development is the discovery of molecular subtypes within follicular lymphoma, a common form of non-Hodgkin lymphoma. Using advanced techniques like whole genome sequencing, researchers have identified 3 distinct genetic subgroups. Each subgroup has unique genetic mutations and immune environments, which means patients can now receive treatments tailored to their specific subtype. For example, patients with the C2 subtype might benefit from drugs that target BCL2 and EJ2, while others with different subtypes could respond better to inhibitors targeting pi3 degrees Kelvin, IRF4, or BTK pathways. This shift toward precision medicine allows for more effective and personalized treatment plans. In diffuse large B-cell lymphoma, another common type, scientists are using single-cell RNA sequencing alongside traditional bulk transcriptomics. This approach helps identify gene signatures linked to immune cells called regulatory T cells, which can predict how well a patient might respond to treatment. It also reveals genetic mutations associated with high or low risk, giving doctors better tools to forecast disease progression. Understanding the tumor microenvironment, the surrounding immune and tissue cells, has become crucial in predicting how the disease will behave and how patients might respond to therapies like CAR T-cell treatment. Proteomics, the study of proteins, is also advancing rapidly. By analyzing proteins at a large scale and at the level of individual cells, researchers can discover new biomarkers that help classify lymphoma more accurately. Combining proteomics with genomics and transcriptomics, known as multi-omics, provides a comprehensive picture of the disease. While these techniques are promising, translating them into everyday clinical practice still faces challenges, such as variability in samples and the need for extensive validation. Another exciting area is liquid biopsy, which involves analyzing circulating tumor DNA in the blood. This non-invasive method allows for real-time monitoring of genetic changes in lymphoma without needing tissue biopsies. It can detect mutations, track how the cancer evolves during treatment, and identify minimal residual disease, small amounts of cancer cells that remains after therapy. This helps doctors predict relapse early and adapt treatment strategies accordingly, improving patient outcomes. Single-cell epigenomic techniques, like SC-ADAC-seq, are shedding light on how lymphoma cells develop resistance to drugs. These methods reveal changes in DNA methylation and histone modifications that help cancer cells evade treatment. Understanding these mechanisms can lead to better timing of therapies and strategies to prevent resistance from developing in the first place. Finally, genetic profiling has directly influenced the development of targeted therapies. For instance, drugs like ibrutinib inhibit specific molecules in the B-cell receptor pathway, which is often overactive in lymphoma. By understanding the genetic drivers of each patient's disease, clinicians can choose treatments that are more likely to be effective, reducing unnecessary side effects and improving the chances of remission. In summary, breakthroughs in lymphoma genetic profiling are transforming how we understand and treat these cancers. From identifying distinct molecular subtypes to using liquid biopsies for ongoing monitoring, these advances are paving the way for more personalized and effective care. Always remember, if you or someone you know is affected by lymphoma, consulting with healthcare professionals is essential to determine the best approach based on individual genetic and clinical factors. These scientific strides are helping us move closer to a future where every patient receives treatment tailored specifically to their unique disease profile.